defense, right? And the biggest defense, hopefully, that nobody gets cancer, nobody gets autoimmune diseases. Most of the people, hopefully, God willing, would not have it. But infections, you'll always get it. So this means that our immune system is playing at all time, because we are exposed to infection. Infection is actually the number one killer everywhere in the world if you look at diseases. Now, the immune system, again the same story. If you look at the major concern we have that you will see is bacterial infection. I say major because we can do something, and I'm pretty sure in your pharmacy practice you're going to use antibiotics. But after COVID-19, I tend to change a little bit, saying viral infection, but again, I don't know. I haven't myself. I've been prescribed antiviral drugs, but I hate to take them. So very rarely people take antiviral drugs, but they've been prescribed these days. And industry is spending a lot of time on antiviral drugs. But normally most of the people do not. So they still want immune system to tackle with that. But you never know. Things may change. So the major concern is infections. Now you can see that we basically have two major types, gram-positive and gram-negative. We discussed that in microbiology anyway. And what immune system actually sees in this case is, again, the outer picture. Immune system looks like outer feature of that, because it has eyes. T cells has an eyes symbolically, and B cells. So they will look at the outermost layer, which is like a cell wall. So if we look at a little bit of recap of cell wall, so you can see, remember the idea I told you that carbohydrates, proteins, and fats, and nucleic acids, they are like antigens. They act like an antigens. So they have this fatty layer, teichoic acid, carbohydrates, and protein in the cell wall. So immune system is going to see that. Okay? And then again, the other important thing is that the immune system is going to recognize that and kill it. But immune system has to be careful. So if it's going to kill, like shoot them, there's going to be a lot of debris. So it does, like, I would say, a demolition kind of a thing, where everything is done in a very organized way. So if you were to demolish a building, you don't want to just, well, sometimes they do, but most of the time you want to make sure that each thing is separated, right? And each thing is for recycling purposes, for some other purposes. But you can see cell wall, the outermost, had 3 layers, right? And they are going to be broken up and presented to our T cells, and T cells will make antibodies against that. That's like a typical antigen presentation cell. The other thing is that there's a thick layer of protection for gram-positive, and they are resistant to lysis by complement. Complement is another source where we sugarcoat the bacteria with complement proteins and make it palatable, presentable to phagocytes. But this one, again, is resistant to lysis, so we'll have a question over there. What it means is, if you have complement deficiency, you may not be able to deal with gram-positive, kind of immunodeficiency. The other important thing is that the defense against gram-positive is basically opsonin and phagocytic cells, which are neutrophils and macrophages. So they are the major. One of the things that you will see if you have infection, there's a boil, so it forms pus. So what is pus? It's a dead bacteria and dead phagocytic cells like neutrophils and macrophages, because they are like first responders to go and phagocytose them, to kill them. So these are the major, and they have to be opsonized, right? Like sugar-coated. And then you also have to keep in mind that this is a rule over here. Complement does not live gram-positive directly, but provides opsonin and mediators of inflammation that are important for host defense. So complement doesn't directly do that, but it kind of acts like a bigger player to take. So the message for the gram-positive bacteria is that if you're immunodeficient, for example, of complement or monocytes, then you will have a problem of fighting gram-positive bacteria. So you're going to get repeated. You have to ask yourself, you don't want to put somebody on antibiotics forever, because they have their other problems, but this is what we will see. So again, if you do staining, is a peptidoglycan layer. This is like the structure thing. If you compare with gram-negative, you will see that if you remember, gram-negative was like a double sandwich kind of a layer. It's very thick. And in this case, it has an outer membrane which is called lipopolysaccharide, which is endotoxin. It's like a poison. So that is the number one stimulator of immune response, endotoxin. And if you remember microbiology, we talked about endotoxemia. If you get if you see a patient who has gram-positive infection, bacteremia in the blood, or gram-negative, so gram-negative is serious as compared to gram-positive. And both are serious, but gram-negative is more, because it has a polysaccharide portion at the lipid as antigen determinant that counters antigen specific. You don't have to worry about that. But the most important thing is LPS is highly toxic and produce a major response. So if your immune system is not working, you will have endotoxemia, which is still a big player. So you'll see antibiotics against gram-negative as very serious thing, okay? And then again, it also complement system plays a role, antibody specific plays a role, and phagocytic cells like neutrophils and macrophages, they do play a role, okay? So this is like a picture for most of the bacteria. Any questions on that? Okay, let's go to the slide 20. And spirochetes, that's usually got STDs. So if you look at mycobacteria, if you know the structure, it was very complicated. If you remember one of the slides that showed very complex fatty layer. And then again, its cell wall provides strong immune responses during infection. That was the basis of delayed type hypersensitivity for, if you remember, I said tuberculin test. Right? So that was important. And then again, the primary defense mechanism of the immune system is macrophages and cell-mediated immunity. So if you don't have cell-mediated immunity, then you will have tuberculosis. Just guess which is the major disease that people lose cell-mediated immunity? AIDS. You know CD4 cell. So many a time AIDS people have TB as well. They can't fight because the CD4 T cells are deficient. The other are spirochetes. In this case, the most important is syphilis. The bacteria is Treponema pallidum. And then again, you can see that the primary defense against these are complement, specific antibody, and cell-mediated immunity. Again, very high chances for people to have a complement deficiency, and they don't form those specific antibodies that are required for Lyme disease as well, and spirochete infection. Okay, so you can see these are how linked. If you look at the immune mechanisms which are provided by pathogens, so we have, in a bigger picture, we have encapsulated bacteria which have a capsule, and these capsules are polysaccharides. They're like, I would say, like a highly protective layer where it masks the identity of the bacteria. It doesn't want immune system to look at it. And the other thing is that you can see, if I tell you the capsules interfere with complement system, it's a very weakly immunogenic, right? So this means that you may have infection and your immune system would not know. You don't want that. So our immune system is designed to fight infection, and the bacteria are designed to resist infection. This is being caught up by the immune system. So this is how the fight goes on. And again, you can see there are special type of cells which are spleen and liver. They actually are critical for protection against capsulated bacteria. So if you remove a spleen or you have a problem with the liver, you are exposed to encapsulated bacteria. Okay? And then again, if you look at the mechanism is pretty much the same for most of them. We have opsonins like that for phagocytosis and neutrophils and macrophages, which are also called phagocytes. So they are going to go phagocytosis. The other important thing is that sometimes these bacteria, like gram-negative bacteria, they release toxins. There are two types of toxins. One called exotoxins, the other are endotoxin. Right? Endotoxins are more dangerous. But in this case, the toxins are proteins, and they produce physiological effects at minute concentration. And then again, in order to protect you from that, you have IgG. But again, IgA antibody is also important. And then many a time we usually, like for example, tetanus. You get tetanus toxide, which is an antibody against tetanus. But tetanus is a bacteria that releases a toxin, right? So any bacteria that releases a toxin is going to spread as a poison. So we have to have specific antibodies for that. That's why you get vaccinated for some of the bacterial infections. Okay. So this is also how the immune mechanisms are involved when you are infected by either a capsulated bacteria or a toxin. And then again, you can see over here, we talked about endotoxin on the gram-negative, but they are exotoxin. Exotoxins are those toxins which are excreted out of the bacteria, and endotoxin is a component of the bacteria. That makes sense? So these are two important things that you have to keep in mind. Any questions on that? Okay. Let's move on to slide 37. Give you an idea, because all bacteria are foreign. All bacteria are foreign, right? And I was looking at some of the exams, because I must have told many a time, I said, write it up in practices that all immunogens are antigens, but all antigens are not immunogens. So that should stick in your mind. But I still see some people, I don't know how many, one or two still made a mistake on that. So that should be very clear on your mind. So antigen is anything that provokes a response, right? And then again, some of the antigens are super antigens. They're gonna be like Superman, do extraordinary things. That's why we have super. And sometimes the antigens vary. Right? It depends upon which antigen we're talking about. Now in this case, if you have gram-positive infection, which is called by staphylococci, which is a gram-positive bacteria, so you will have a disease called toxic shock syndrome. Does anybody remember what I taught in toxic shock syndrome? I gave a story as well. Staphylococcus infection, and that they took out, initially in the first few years of that. So that was the toxic shock syndrome. Then there was a picture of a baby which had this scarlet. I actually showed two pictures. One of the young girl which has peeled off the skin, and I told you it looks like a burn. It looks like a burn, but it is a toxin. And then I showed you the little baby which has like scarlet. That happens especially in this case. So what happens in this case, if there is a super antigen, it stimulates large number of T cell, and it takes you like cytokine storm, is exaggerated immune response. Right? That's why super antigen is going to be more. It's just like COVID. So COVID is recognized by immune system, but if the immune system put a response which is like out of proportion, that's also not good. Because you remember I told you it has to be balanced out. If you have too much of response, you're going to get hypersensitive reaction. If you have too little of response, you have energy, no response at all. So it has to be optimal. So this is what happened. Because this super antigen is associated, when you have this toxin release, is going to go hypotension, hypovolemia, organ failure, and death. And B cell super antigens are famous for that. Right? So antigen is there, but this antigen causes a response which is not required, is overreact. Then there are antigenic variation. So what happens is that your body makes, like for example, the question is, why do you get a flu shot every year? How do you then, because the shot that you're gonna get, you're right, it can mutate. The mutation is antigenic variation. So if I get a flu shot, what exactly is the antigen in that? So let me rephrase my question. The question is that, are you getting vaccinated by the virus which is infecting you right now? No. Because it's not possible to do that, to make vaccine. So you are getting from the antigen virus that was in the last season, and you have enough time